Okay, very good evening, my dear friends. And also Ranjit. Today, our student Ranjit from Cheriala, Government Degree College Cheriala, is also joined. A student is very much more interested in the PG entrance botany. And also welcome friends. And also today's eminent resource person, Dr. RANI-CPGET-22, Assistant Professor, Department of Botany, Government Degree College, Autonomous. Madam, please welcome. And Madam, please welcome and start your session. Please share your screen.
Okay, sir. Thank you, sir. Please give me the host, sir.
Okay.
I'm unable to share the screen.
Okay, no problem. Better you have to once try it.
Okay. Now, okay, sir.
Right.
Okay. We'll proceed. Thank you, sir. Thank you very much. Okay, very good evening, students. Yesterday, I will discuss about the introduction of photosynthesis and what is the site of the photosynthesis. Some pigments also we'll discuss yesterday. And today we are going to discuss about the remaining topic, that is generally photosynthesis is divided into two phases. First one is the light phase and the second one is the dark phase. Today we are going to discuss about the light phase. So before entering the topic, I will remind one or two topics, yesterday topics, almost the definition and which pigments are involved in the photosynthesis. So that is chlorophylls, chlorophyll A, B, C, D, E, chlorobium chlorophyll 650, chlorobium chlorophyll 660, bacterial chlorophyll A, bacterial chlorophyll B. So these all are belongs to chlorophylls, Nana. And carotenoids. So in that carotene and xanthophylls also very important for the photosynthetic pigments. And remaining all the phycobillions. So allophycocyanin, phycoerythrin, phycocyanin. So these all are completed yesterday. And absorption spectrum and action spectra also we'll discuss. That is electromagnetic spectrum, visible spectrum, VIBGR. So the 7 colors are involved in the radiation. So wavelengths starting from 350 to 780 nanometers in between that wavelength. So in that almost to the in the action spectrum, the photosynthesis is illustrated in the just the quantum yield curves. Just to okay. So in that two very important bits are there. So one is the red drop and second one is the Emerson enhancement effect. So what is red drop? Red drop means almost to the red color wavelength. So that is we will discuss today, that is PS1 and PS2. So red wavelength of the photosystem. So in that generally the maximum quantum yield occurs in between the 600 to 680 nanometers. So at this wavelength, the wavelength longer than 680 nanometers, there is a sudden drop in the quantum yield. So this phenomenon is known as red drop. So this is first proposed by Emerson and Lewis in 1943. That's why. So the red drop means the wavelength, longer wavelength of the 680 nanometers. So it is involved in the process. So the photosynthetic rate is suddenly dropped. So the sudden drop in the quantum yield, that phenomenon is called as red drop. So likewise, another experiment also they will conduct, Emerson enhancement effect. So the Emerson showed that relatively low photosynthetic efficiency of chlorella. Chlorella is the green algae, Nana. So it is unicellular green algae. The chlorella, they are used in this organism to the yield of the photosynthetic rate. So Emerson first proposed that. Emerson, he showed that the relatively low photosynthetic efficiency of chlorella in the far red light, it can be increased by simultaneous applications of the short wavelength red light. So almost 680, 650 and 700. These all are also, both are the red wavelength spectrums. So they used that far red light and short wavelength of the red light. So in other words, suppose X is the rate of photosynthesis in the one wavelength, long wavelength, and Y is the rate in another wavelength, that is the short wavelength. One is long wavelength and another one is the short wavelength. So for example, we will take that the photo system 1, that is P700, is the long wavelength, and
Excuse me,
second
madam.
one is the,
Madam,
sir?
your slides is not visible.
Okay, sir. Almost I will arrange in the laptop, but it is one or two slides are missing in this way, sir. Just
Okay, no problem, madam. Okay.
Okay, sir. Okay. So the two wavelengths, that is longer wavelength and shorter wavelength. Longer wavelength is the 700 nanometers and shorter wavelength is the 680 nanometers of the wavelength. So these both wavelengths are given simultaneously. It is greater than the sum of the long and short wavelength, X plus Y. So that is called Emerson enhancement effect. So generally, this is very important in entrance point of view. Only one wavelength is used in the photosynthetic. The photosynthetic rate is completely decreased, that is red drop nano. And second one is just when we will use simultaneously two wavelengths. One is longer wavelength and another one is the shorter wavelength. Both will applied at a time. It will they are simultaneously applied, then the complete it is greater than the sum of the A plus B or X plus Y. So that is called Emerson enhancement effect. It is completely applied on the unicellular algae, that is chlorella, green algae. So Emerson showed that the relatively the enhancement or photosynthetic rate increasing of that chlorella. So it is experimentally they are done. So the discovery of Emerson effect, he opened the door in understanding the role played by the chlorophyll A and chlorophyll B. So these both are accessory pigments in the photosynthesis. So yesterday we'll discuss that completely chlorophylls A and B structures. So it is now confirmed that two separate groups of pigments, they are cooperate in the photosynthesis. So these are designated as pigment system or photosystem 1 and 2. So now we will entering into the light reaction completely pigment system 1 and pigment system 2. So the photophosphorylation, it is completely light phase, Nana. As I told you that photosynthesis is completely complete in two steps. Light
Madam, if
phase
possible,
and
rotate the mobile.
Okay. Okay, sir. Sorry. Just.
And
Okay, sir.
please enlarge the this slides. If yes, ma'am. Okay.
Okay, sir.
Yes, madam. Yes, proceed.
Okay. Photophosphorylation, it consists of two pigments to excite, that is called PS1, photosystem 1 or pigment system 1, and second one is the PS2. So that is PS2, photosystem 2 or pigment system 2. So generally, now we are coming to the PS1. PS1, it is contains the chlorophyll A. So please keen observe here. Completely the pigment system, both PS1 and PS2, the pigment system 1 is activated by light with wavelength longer than the 680 nanometers. That means 700. So PS1 is better excited by light at about 700 nanometers of the wavelength. Okay, PS1. So the PS1 or photosystem, it is comprised pigment protein complexes. Both PS1 and PS2 is the protein complexes they are associated with the reaction centers. Okay, so the reaction centers are a small percentage of chlorophyll A molecules. They are capable of the transferring electrons. So in the PS1, it contains the chlorophyll A, small amount of the chlorophyll B, beta carotene, and some of the chlorophyll molecules also. They are in a special chlorophyll. It absorb light at 700 nanometers of the wavelength. So you can observe here PS1 is better excited by light at about 700 nanometers and is thus sometimes called P700 reaction center or P700 nanometers of the wavelengths. Okay, completely it includes the chlorophyll A, small amount of chlorophyll B, beta carotene and proteins. Okay, so in this generally it is called the P700, the reaction center to which all surrounding chlorophyll A, B, beta carotene to the photosystem transfer their energy. And pigment system 1, they contains the iron sulfur protein. So it is very important. It contains P700 is the reaction center, Nana. So P700, the system, it contains the iron sulfur proteins. Completely primary electron acceptor of the PS1 is iron sulfur proteins. So which are the primary acceptors of the electrons? They are transferred from the reaction center. So transferred from the reaction center means P700 is the reaction center. So the electrons are transferred from reaction center to the primary electron acceptor, that is the iron sulfur proteins. So PS1 generates a strong reductant capable of the reducing NADP plus and a weak oxidant. So these all are just we'll discuss in coming. And second one is the PS2. PS2 is pigment system 2. It contains the chlorophyll A. So small amount of chlorophyll A and more chlorophyll B. So complete quite opposite, Nana. PS1 is more chlorophyll A, small chlorophyll B. But PS2 is the completely chlorophyll A, small chlorophyll A, more chlorophyll B. And beta carotenes, proteins also have in this PS2. So it is located between the closely stacked grana. So yesterday I discussed that the structure of chloroplast, and completely PS2 is located between the closely grana and it is not in direct contact with the stoma. So PS2 reaction center is P680 nanometers. P700 is the PS1 reaction center. P680 is the PS2 reaction center. So it is also contain the primary electron acceptor, that is the phyophytin. As per same, PS1 is the primary electron acceptor in the PS1 is the iron sulfur protein, but in PS2 is the primary electron acceptor is the phyophytin. So phyophytin, it is a molecule of chlorophyll without magnesium ion, Mg2+. So it is P80 and P680 and proteins are bound to quinone. So Q cycle or Q ring, generally this is phyophytin completely, it is a protein, they are bound to the quinone circle. So it is also contains some proteins bound to the manganese, they are called the manganese protein. This is the PS1 and PS2. So generally now we are entering into that electrons are in the photosynthesis process, the phosphorylation of ADP to ATP using the energy of sunlight, that is called photophosphorylation, Nana. Simply ADP plus inorganic phosphate gives rise to ATP. So adenosine diphosphate adding with inorganic phosphate and they are converted as a ATP, adenosine triphosphate. So in the photosynthesis, almost the phosphorylation of ADP to ATP using some using the energy of sunlight, that is called photophosphorylation. In the photophosphorylation, the light energy is used to create a high energy electron donor and a lower energy electron acceptor. So in this completely, as I told you that photosynthesis is light phase, dark phase. So in the light phase, they are accumulate some of the simulation of energy in the form of ATP and NADPH2. So that's why the Arnon, first time Arnon demonstrated that the isolated chloroplast, they are produce ATP in the presence of light. So they termed the process of ATP synthesis in the chloroplast as photosynthetic phosphorylation or photophosphorylation. Okay, so completely photo means in the presence of light. Phosphorylation means ADP combined with inorganic phosphate to convert as ATP. So that is the phosphorylation. Okay, it is first demonstrated by Arnon, ARNON, Arnon. So Arnon demonstrated the chloroplast, they produce the ATP in the presence of light. So that is termed the process of ATP synthesis. Or so they are also known as the photophosphorylation or photosynthetic phosphorylation. Okay, so completely in the photophosphorylation, light energy is used to create a high energy electron donor and a lower energy electron acceptor. So the electrons then move spontaneously from donor to acceptor through an electron transport chain. Completely. So before entering the ETS system, we'll discuss later. First, we are entering into the types of photophosphorylation. So there are two types of photophosphorylation. First one is the cyclic photophosphorylation and the second one is the non-cyclic photophosphorylation. So in this, cyclic, first we will discuss about that cyclic photophosphorylation. Okay, so the cyclic photophosphorylation, it occurs in chloroplast. So it is illuminated with far red light. Cyclic photophosphorylation occurs in the chloroplast, illuminated with the far red light. So under this condition, only photosystem 1 is activated, PS1. So cyclic electron flow involves only one pigment system, that is the PS1. Okay, so the cyclic photophosphorylation is very important because it provides a source of ATP that is independent of the production of the NADPH. So electrons passed from P700 to pyridoxine, they do not continue to NADP plus but move back through the cytochrome BF complex to the plastocyanin. So you have little knowledge on this almost in the 4th semester. It is very important in theory examination also. So just I will give in detail of the cyclic and non-cyclic photophosphorylation. Generally, just I will share this is the cyclic photophosphorylation. So cyclic photophosphorylation is very important. It provides a source of ATP. So generally, the cyclic photophosphorylation start with when PS1 is activated. You can observe here, Nana. So, this is the PS1 is activated by 1 photon, almost the green color photosystem 1. It is observed the light or waves. So that is in the form of photons. So when PS1 is activated by 1 photon, the excited electron is that is lifted from its reaction center P700. So we know that PS1 reaction center is the P700 nanometers of the wavelength. So that is red light. So it is completely lifted from its reaction center P700. The electron passes through a chain of electron carriers with increasing the reduction potential. So that means the first carrier is designated as generally completely photon. You can observe here the wavelengths. So when observe the light, so rays, so it is undergo the excited state, it will gives the electrons. So the electrons are accepted by the iron sulfur protein. So first primary acceptor of the PS1 is the iron sulfur protein. So instead of the electrons finally proceeds through ferredoxin A, B, so it contains the iron sulfur. Instead of being passed on onto the electrons, they are returns from reduced ferredoxin by cytochrome B6. You can observe here P700 to electrons are moved to the primary electron acceptor. E means iron sulfur protein, that is the primary electron acceptor of the PS1. PS1. So from the iron sulfur protein, the electrons move to the plastoquinone through the ferredoxin. So the plastoquinone becomes reduced to that means almost to PQ to QH2. So it is reduction and oxidation states also they are observing in that state. So the protons translocated through plastoquinone pool, they are released into the lumen of the thylakoid membrane. So electrons are subsequently transferred to the plastocyanin. So PQ2 cytochrome complex in between. So the electrons are moved from PQ to PC. PC means plastocyanin. So thus the photo oxidized P700 that is accepts the electron from reduced plastocyanin. So the electrons lost by P700 is cycled back to P700, causing the accumulation of protons inside the thylakoid. So it results in setting up a proton gradient and the subsequent formation of the ATP. In this cyclic photophosphorylation, the formation of ATP by this completely cyclic electron transport is called cyclic photophosphorylation. That means when it starts from the photosystem 1 reaction center to electrons move through the channel of iron sulfur protein, plastoquinone, plastocyanin, and again back to the reaction center P700. So that's why the formation of ATP by the cyclic electron transport. So that is called cyclic photophosphorylation. So what is the use of cyclic photophosphorylation? Generally, during the cyclic photophosphorylation, ATP is produced. So ATP produced via proton motive force. So see here, ATP is almost it is produced via proton motive force. So in the cyclic photophosphorylation, no oxygen is released and NADP also it is not reduced in this cycle. So that's why it is only the assimilation power, or it is give the ATPs in between the plastocyanin to cytochrome and plastoquinone to plastocyanin. So completely the formation of ATP by the cyclic electron transport, that is called cyclic photophosphorylation. You can observe here cyclic electron flow, it produces the ATP but not NADPH2 and but they no oxygen also released in this cycle. So at some point the plant cells do not require much NADPH for biosynthesis process, but still require the ATP for other biological process. So it needs to vary the ratio of NADPH and ATP formed. So this is done by an alternative path of light induced electron flow. So electron flow, electron transport system, or it is called cyclic electron flow. In completely, so the cyclic electron transport, the in the end completion of the cyclic will get some ATPs So and next one is the non-cyclic photophosphorylation. Sorry for the interruption. Okay, non-cyclic photophosphorylation. The non-cyclic, you know here. So cycle means almost starting point to ending point. The electrons move through different electron carriers or different electron transporters. They are reached finally the starting point, that is the cyclic. Non-cyclic means, so it is first proposed by Hill and Bendall, Nana. Hill and Bendall in 1961. So they proposed the scheme of photosynthetic electron transport. It involves the both photosystem 1 and 2. So in cyclic only photosystem 1 only participate involves in the cyclic, but in the non-cyclic PS1 and PS2 both will acting in series. They connected by electron transport mediators. So it's look like a drawing this scheme on a redox scale. It gives rise to the letter Z. So you can observe here N or Z. So hence this scheme is often known as the Z scheme. So cycle means generally cyclic lo manaki ekkada start ayite akkadike end avutundi, but in non-cyclic it involves the two systems PS1 and PS2 both will involves in this non-cyclic. So in as drawing this scheme in the redox scale, so it gives rise the Z letter or N letter reverse. So letter, and hence the scheme also often known as the Z scheme. So the process, it begins with the absorption of light energy by light harvesting complex. Just to, I'll share another. Wait. Photosystem 1 and 2. So the light reaction, Hill reaction. So the reaction center photosystem 1 is completed. The reaction center chlorophyll of photosystem 2, it observed the maximum at 680 nanometers. The reaction center of the PS2 is the 680 nanometers. So its reaction center chlorophylls and associated electron transport proteins is located predominantly in the stacked regions of the grana lamellae. So at a wavelength greater than 680 nanometers, this is what red drop, as I told you. So only one system is involved in that stage. So that is the red drop. Photosystem 1 contains the large amount of chlorophyll A and a small amount of chlorophyll B and some beta carotenes. These all are completed, Nana. So in the light reaction, just to photophosphorylation, now we are entering into the non-cyclic photophosphorylation. So this process begins with the absorption of light energy by light harvesting complex. So it is transferred to P700. So both systems are involved. Photosystem 1, photosystem 2, in between redox chain. So energy of molecules also you can observe here. So in this system, the first one is the PS1. So it is transferred to P700 by resonance transfer. So the light excited P700 expels the electrons. In the first one, this is the PS1 P700 reaction center. So from the right side, the PS1 is participated first. After that, the electrons, they are passed through a chain of intermediate electron carriers to a soluble pyridoxine present in the stroma. So the first carrier, they are look like appears to be we'll treat it as A0. First carrier is A0. So it appears to be a chlorophyll A, whereas the second carrier is A1. So it is probably a phylloquinone or iron sulfur. Just to, as per your knowledge, PS1, P700 is the reaction center. The electrons are moved from the reaction center to primary electron acceptor is the iron sulfur protein. So after the iron sulfur protein, almost the electron finally proceeds through the 3 membrane bound ferridoxin. So that is completely Q. Or in this way, they are contained the iron sulfur and reaches the soluble ferridoxin that is located in the stroma. So the redox chain or stroma, and they are reduced ferridoxin in turns they are reduced to NADP+. So this is very important. Almost in the right side, the photosystem 1 indicating as the photosystem 1 electrons move through the primary electron acceptor to just this one, Nana, ferridoxin. So they are iron sulfur protein and reaches the soluble ferridoxin FD. So that is located in the stroma right side. So they are reduced ferridoxin in turn they are reduced to NADP+. So FD bound or FD soluble. So here it is indicating as FD bound and FD soluble. So the ferridoxin, they located in the stroma, they are reduced to NADP+. For reduction of 1 molecule of NADP, 2 electrons are required, and these 2 electrons are provided by ferridoxin 1 at a time. Can you catch this point? Almost here is 2 PS1, PS2 both will involves in this non-cyclic. So start with PS1, the reaction center P700. So the electrons are move from reaction center to primary electron acceptor. After the primary electron acceptor is iron sulfur protein. So the electrons are moved from iron sulfur protein to ferridoxin. So ferridoxin to almost the ferridoxin, it is reduced to NADP+. NADP+. So for reduction of 1 molecule of NADP, 2 electrons are required. So almost each time 2 electrons move through the electron transporter. Almost so reduction generally 2 electrons are required, and these 2 electrons are provided by the ferridoxin, 1 ferridoxin at a time. So the reduction is catalyzed by FAD, that means ferridoxin NADP reductase is involved. So it FAD containing the ferridoxin NADP reductase enzyme. Okay, so this ferridoxin NADP, it reductase that is apparently involved in matching the 1 electron chemistry of ferridoxin to the 2 electron chemistry of the NADP. Catch this point, Nana. Almost ఇప్పుడు 2 electrons photon నుంచి primary electron acceptor కు వచ్చినాయి iron sulfur protein నుంచి FD radiation stage కు వచ్చిన తర్వాత ఒక NADP reduce కావాలి అంటే 2 electrons అవసరం. So ఆ 2 electrons is transport in every time, but these 2 electrons are provided by ferridoxin at a time. So they are catalyzed by which enzyme? FAD. So that means ferridoxin NADP reductase enzyme is essential for the NADP H2. Okay, so this enzyme ferridoxin NADP reductase, so that is involved in matching the 1 electron of the ferridoxin to 2 electron of the NADP. Okay, since the expelled electron, they are ends up in reducing NADP+, so it results in electron deficiency in the P700. Almost 2 electrons are passed from the reaction center. So finally PS1 photosystem 1 is, it is electron deficiency also we will observe in the PS1. So it is fulfilled through the photolysis of the water. Just to discuss that. So commonly it is the P700 reaction center is referred to as a whole. So this deficiency,
here
it
one
is
of the most
filled
important, madam. Here one of the most important, DCMU. DCMU, one of the most important chemical components.
Oh, okay. Okay, sir.
which is cut down the electrons from
Okay.
move from photosystem 2 to photosystem 1. So the
One.
this important